Ladies and gentlemen, on behalf of InVivoScribe, we welcome you to this webinar organized by AMP, the Association for Molecular Pathology. Our program's title today is Explore the Benefits, Considerations, and Utility of NGS-B and T-cell clonality testing. We encourage you to ask questions in real time using the chat feature so that time permits we can address them at the end of the presentations. I'm very pleased to introduce our speakers for today. Our first speaker is Dr. B. Melody Zhang, medical doctor, master's in science, and fellow of the College of American Pathologists and American College of Histocompatibility and Immunogenetics. She is the clinical assistant professor and assistant director of the Histocompatibility and Immunogenetics Laboratory at Stanford University School of Medicine. Dr. Zhang holds American Board of Pathology subspecialty board certifications in clinical pathology and molecular genetic pathology. Dr. Zhang completed clinical pathology residency, molecular genetic pathology, as well as clinical genomics fellowships and histocompatibility and immunogenetics training at Stanford. She is an attending pathologist at the Molecular Pathology Service and assistant director of the HLA lab. She is also certified by the American Society for Histocompatibility and Immunogenetics. Her research interests include genetic and genomic testing for malignant and non-malignant hematologic disorders, NGS-based T-cell receptor testing, and MRD diagnostic testing, as well as molecular testing for solid organ transplantation, HLA-related disease association, and pharmacogenetic testing. Dr. Zhang's presentation is titled Comparison of T-cell receptor gene rearrangement detection with capillary electrophoresis versus NGS. Our second speaker today will be Dr. Bijal Parikh, medical doctor, PhD. He is assistant professor of pathology and immunology, medical director of the Molecular Diagnostics Laboratory, associate medical director of the Molecular Infectious Disease Laboratory, and associate medical director of the HLA Laboratory at the Washington University School of Medicine. Dr. Parikh holds American Board of Pathology subspecialty board certifications in clinical pathology and molecular genetic pathology. He is also highly recognized, having received the American Association for Clinical Chemistry Outstanding Research Award in Molecular Pathology and Pharmacogenomics, and the Academy of Clinical Laboratory Physicians and Scientists Paul E. Stanford Young Investigator Award. He also received the Washington University Department of Pathology and Immunology Chair's Special Recognition Award, COVID Clinical Hero. Dr. Parikh completed clinical pathology residency and molecular genetic pathology fellowships at the University of Washington in St. Louis, Missouri. He is an attending pathologist in St. Louis and is affiliated with the Barnes Jewish Hospital. His main research interests include exploring mechanisms governing host pathogen interactions with ongoing research projects around the development of a murine cytomegalovirus model to study innate immune control by natural killer cells and trafficking of virus during neonatal infection. Today, Dr. Parikh's presentation is titled Characteristics and Utility of NGS-based IGH clonality and IGHV somatic hypermutation detection. I am now pleased to turn the time over to our first presenter, Dr. Melody Zhang.
Thank you for the introduction, Paul. Hi, everyone. It is a great pleasure for me to have this opportunity today to share my experience on NGS-based T-cell receptor gene rearrangement detection and its comparison with capillary electrophoresis-based methods. There are three main learning objectives for this first part of the webinar: to understand the advantages and limitations of PCR-CE-based versus NGS-based T-cell receptor gene rearrangement assay used to detect the T-cell clonality, to learn about the characteristics of the LymphoTrack TRG assay as demonstrated in a research study at our institution, to learn about the considerations on transitioning from PCR-CE-based to NGS-based T-cell receptor gene rearrangement detection. Here is the outline of the topics that I will cover in my presentation. First, I will briefly review the structure and the genetics of immunoglobulin and the T-cell receptor, followed by the T-cell development and T-cell gene rearrangement. Then I will talk about the molecular testing methods of T-cell receptor rearrangement. After that, I will go over the main performance characteristics of NGS-based TRG assay. In the end, I will use some examples to demonstrate the considerations in transitioning from PCR-CE-based to NGS-based TCR gene rearrangement detection. Let's first review the structure of IG and TCR. B and T lymphocytes are characterized by their antigen-specific receptors, which are the primary effector molecules of the adaptive immune system. For B cells, they are the membrane-bound form of the immunoglobulin as the main component of the B cell receptor, and the soluble form of IG secreted by plasma cells. Both forms of IG have the identical Y-shaped structure, composed of 2 heavy chains and 2 light chains. On the other hand, T-cell receptors are heterodimeric cell surface molecules responsible for recognizing antigens presented to T cells in the context of MHC molecules. In the circulation, majority, about 95% of T cells, express alpha-beta chains, and a minority of T cells express gamma-delta chains as a part of the TCR complex. Gamma-delta T cells are a dominant subtype of T cells found in epithelial tissues. Both the TCR and IG molecules consist of variable and constant regions. Each lymphocyte receptor has a unique antigen specificity determined by the structure of antigen binding site, primarily from the variable amino acid sequences. At the genetic level, there is a complex and elegant mechanism to generate the remarkably diverse lymphocyte antigen receptor repertoire needed to respond to a great variety of antigens. Due to the extremely high level of diversity, the receptor chain variants cannot be encoded in full in the genome. Instead, they're encoded in the germline genome in several pieces as variable V, diversity D, and adjoining J gene segments, with each type of gene segments present in multiple copies. These gene segments are assembled in the developing lymphocyte by random somatic DNA recombination to form a complete variable region sequence. This mechanism is known as gene rearrangement. V and J segments are present in all antigen receptor genes, and D segment is present only in some receptor genes. The large number of possible recombinations of different segments account for majority of the receptor repertoire diversity. In addition, there are non-templated insertions and deletions of random nucleotides at the junctional regions. This occurs early in lymphoid ontogeny in both B and T cells, especially they are in tandem with the rearrangement process and further increasing repertoire diversity. The rearrangement process for TCR beta, or the TRB gene, is depicted in this figure. It usually starts with a D to J rearrangement, followed by a V to DJ rearrangement. The same process applies to TCR delta, or the TRD gene. TCR alpha, the TRA, and the TCR gamma, the TRG genes do not contain D gene segments, and their rearrangements involve direct V to J recombinations. T cells develop from common lymphoid progenitor cells that seed the thymus from the fetal bone marrow or liver. The differentiating T cells in the thymus are either double negative, double positive, or single positive for the expression of CD4 and the CD8 cell surface antigens. During the double negative stage, T cells can be subdivided into 4 subsets, DN1 to 4, based on the rearrangement of their TCR genes and the expression of surface antigens. TCR gene rearrangements are complex and generally follow a hierarchical order. First, the TRD genes rearrange, then the TRG genes, potentially resulting in TCR gamma delta expression, or followed by further TRB gene rearrangement and TRD deletion, with a subsequent TRA gene rearrangement, potentially followed by TCR alpha beta expression. The estimated numbers of functional and rearrangeable human V, D, and J gene involved in the TCR IG gene rearrangements are shown in this table, which is adapted from the landmark paper by Zhang et al. In 2003. The many different combinations of V, D, and J genes represent the combinatorial repertoire, which is estimated to be 3 million for TCR alpha beta molecules and about 5,000 for TCR gamma delta molecules. So what molecular diagnostic methods can we use to test for TCR rearrangements in the laboratory? Southern blotting was used in the past, and the method currently used by majority of molecular diagnostic laboratories is PCR and capillary electrophoresis based. The PCR amplicons of target genes generated using TCR-specific primers undergo capillary electrophoresis to be separated by size. The interpretation is based on the fragment size distribution pattern, which is semi-quantitative. The representative monoclonal and polyclonal patterns are shown on this slide. Compared to Southern blotting, PCR-based method is faster, more accurate, and sensitive, with no need of restriction enzyme digestion, and requires only small amounts of DNA as template. In addition, PCR can also amplify partially degraded DNA. Therefore, PCR-based techniques can be used to analyze small biopsies, such as skin biopsies, or formalin-fixed, paraffin-embedded samples, which generally yield low-quality DNA. The widely used qualitative PCR-CE assays are suitable for clonality assessment and do not allow for precise minimal or measurable residual disease analyses. With the advancements in sequencing technology, next-generation sequencing-based TCR gene rearrangement analyses has emerged and been gaining popularity. Compared to the PCR capillary electrophoresis-based method, NGS-based method can provide the fingerprint characterization of each clonal type, as shown in the example here. They are the specific rearranged TCR gene sequence, as well as the quantitative sequencing count information. It can improve the test sensitivity and the specificity compared to PCR-based methods. It also has the advantages of being feasible for accurate correlation with disease clones from multiple sites and easy to track disease clone longitudinally. The test can be readily performed on benchtop sequencers with capacity to multiplex several samples and different gene targets in the same run. The representative clonal and polyclonal NGS-based TRG test results are shown on this slide, along with the initial PCR CE results of the same samples. On the left, we can see two predominant peaks detected within the V1 to 8 size range on CE. Correspondingly, two predominant clonal types utilizing the V gamma 4 J gamma 1 2 gene segments are detected by the NGS-based assay at frequencies of 32.5% and 32.3%, respectively. fragment length analysis was compiled by tabulating read counts and read lengths. The tabulated read counts of all sizes were plotted as a histogram to show the size distribution of each sample. And as we can see, it has a similar pattern of two predominant clonal types as what we see in CE. On the right is a sample with a polyclonal pattern. PCR-CE showed Gaussian distribution. NGS method showed no predominant clonal type. And the highest one is at 0.89% of total reads. And the NGS rates visualization by size is also consistent with a normal distribution pattern. Our lab performed a comparison study of the NGS-based versus PCR-CE-based TRG rearrangement assays. A total of 101 specimens were included in this study. 46 of these are FFPE specimens, 48 are peripheral blood specimens, and 7 are bone marrow aspirates. And initial PCR-CE-based and a later NGS-based TRG rearrangement testing were performed on all these specimens. The analytical sensitivity of the NGS assay was determined by assessing the detection of 2 expected clonal rearrangements in serially diluted clonal control DNA with polyclonal control DNA across 2 dilution ranges, 0.1 to 10% and 0.1 to 5%. The percentages of total reads detected of the 2 known clonal rearrangements are shown on the left. Both dilution ranges showed excellent linearity with Pearson correlation coefficients of greater than 0.97. Both clonal types of the clonal control DNA was also reproducibly detected at dilutions as low as 0.1%. The polyclonal background level was estimated to be 0.5% of total reads based on the mean of the 3rd most predominant clonal type across all dilutions, since the control DNA is known to have only 2 predominant clonal types. The calculated signal-to-background ratio of each respective dilution study are shown to the right of each panel. Based on the signal-to-background ratio requirement of 5, which is defined in the interpretation criteria, the lower limit of detection for clonality was conservatively determined to be 5% clonal cells, which is equivalent to at least 2.5% of allelic fraction of predominant clones in a specimen. The dashed line here indicates the required signal-to-background ratio of 5, and the data is expressed as mean plus minus standard error of the mean. This table shows the detailed results of the 2 known clonal rearrangements in the serial dilutions and the corresponding signal-to-noise ratios. At 5% of clonal cells, we're able to confidently distinguish clonal signals from the background signal. This multi-alignment shows that one of the tumor clonal type sequence identified at different concentrations in the serial dilution studies. And as we can see, they're 100% identical in the sequence. We also assessed the number of sequencing reads detected in the no template control samples of multiple NGS runs. The read counts ranged from 500 to 1100. Compared to the total reads of the runs, the percentage of reads in no template control is in the range of 0.005 to 0.012%. Majority of samples have total read count over 500,000. So on the per sample basis, the limit of blank is approximately 0.002. The analytical specificity was assessed by testing 40 CE negative samples from a variety of sample types, including 19 FFPE, 17 peripheral blood, and 4 bone marrow aspirate specimens. 39 out of the 40 samples are in concordance. The only discordant case had 3 prominent peaks and was called oligoclonal by CE. Both peaks from the V gamma 1 to 8 were called by NGS as predominant clonal types, and the third one, V gamma 10 clone, was ranked number 3, but was not close to the interpretation cutoff for the NGS to call it as a predominant clone. Therefore, NGS was interpreted as positive. So the intra-run precision was assessed with the clonal frequencies of the top 2 clones for 6 positive samples in triplicates within the same run. The result summary is shown in the table here. As we can see that the intra-run precision was dependent on the particular rearrangement, with a coefficient of variations ranging from 1.1 to 10% for rearrangements not involving J gamma P, and CVs ranging from 27.2 to 48.5 for rearrangements involving J gamma P. Inter-run precision was assessed with the clonal frequencies of the top 2 clones for 5 positive samples across 3 runs. The inter-run precision also seems to depend on the particular rearrangement, with the CVs ranging from 1 to 11.8% for rearrangements not involving J gamma P, and 22.4 for the one involving J gamma P, the one sample we have. This table summarizes the NGS results and how they compare with the CE results. Using the clonality interpretation criteria that I will introduce in a later slide, there was a concordance between NGS and the CE results in 76 out of the total of 101 samples, yielding an overall analytical accuracy of 75%. Of the 76 concordant samples, 41 were clonal, 7 were oligoclonal, and 28 were polyclonal by both methods. For the 41 clonal samples detected by both CE and NGS methods, the predominant clonal type sequences accounted for 2.7% to 65% of total reads and ranged from 5 to 544-fold of the polyclonal background. So on evaluation of the accuracy of clonal versus non-clonal calls, the non-clonal calls included both oligoclonal and polyclonal ones. There was concordance between NGS and the CE in 84 of the samples, yielding an overall analytical accuracy of making clonal calls 83%. The analytical specificity for the NGS assay using the 2-tube-based CE testing results as the reference method for determining clonality was 100%. To further compare the CE and NGS results and investigate possible reasons for discordance, the V and the J genes identified by both methods were examined. The VJ gene identities of the clonal sequences determined by NGS, which is defined as the clones with a frequency at least 2.5%, were concordant with inferred V gene peaks by CE method for 83% of the samples in the study overall. And on stratification by different V and J family genes identified on CE, the concordance rates with NGS ranged from 73 to 95%, with V gamma 11 being the highest and the V gamma 9 being the lowest. The sensitivity values with the CE as the gold standard of the NGS assay was calculated to be in the range of 10 to 96%, with V-gamma 1 to 8 being the highest and the V-gamma 9 being the lowest. Lower sensitivity was correlated with a lower mean V gene usage values, and I will show more data on the next slide. The specificity values were generally good in the range of 81 to 100%. Concordance for J-gamma P could not be assessed because it was not included in the primer sets of the two-tube CE assay. The overall V and the J gene usage by the NGS assay are summarized in this table. As we can see, that the mean usage of V-gamma 9 and V-gamma 11 are lower as compared to others, and they correlated with the lower sensitivities. We also performed the TRG repertoire diversity assessment of the NGS data. The figure on this slide shows the mean Simpson's index of diversity and the mean Shannon's index of equitability of samples determined to be polyclonal, oligoclonal, or clonal by NGS. While the differences in the means of the two indices between polyclonal and clonal samples were significant, only the differences in the mean Shannon index between polyclonal and oligoclonal samples were significant. Our study utilized the unique ability of NGS to assess the sample, the TRG repertoire, and to demonstrate that calculations of diversity may be useful supplemental metrics in the evaluation of clonality. This pie chart shows the demographics of the study population. The clinical pathologic diagnoses of the subjects included in the study are shown over here. And the performance of the CE and NGS assays in the context of the T-cell malignancy and the non-T-cell malignancy is summarized in the table on the right. The NGS assay had a clinical sensitivity of 73%, specificity of 91%, and overall accuracy of 82% based on the current interpretation criteria. Overall, the NGS assay using the existing interpretation criteria was highly concordant with the CE assay with accuracy of 83% and a highly specific 100% in determining clonality. And among the specimen types in the study, the majority of cases showing discordant NGS and CE results were peripheral blood specimens, and that's 17 out of the 25 total. And of the 25 discordant cases, 13 were interpreted as a clonal by CE, but were called as a polyclonal by NGS. And of these 13, 7 showed no clinical pathologic evidence of any hematolymphoid malignancy, suggesting that CE results might have been false positives. So before I show some examples of discordant cases, it is helpful to first give an overview of the design of the PCR CE, as well as the NGS-based TRG assays. In the two-tube design of the TRG CE assay, J-gamma P was excluded in the primer mix to reduce the risk of falsely identifying canonical rearrangements as clonal products, although J-gamma P rearrangements are seen in about 3% of T-cell neoplasms. In addition, with the two-tube design, occasionally polyclonal T lymphocytes demonstrating rare VJ rearrangements may be mistaken for a clonal rearrangement due to the absence of a polyclonal background for that type of rearrangement when separating TRG amplicons into multiple distributions. The NGS assay utilizes a single tube design, which includes the J-gamma P primer and amplifies products in the single size distribution and allow competitive amplification of all gene rearrangements to determine whether a significant population is present. The study interpretation criteria are summarized in this table, and the results are interpreted as a clonal if the most abundant clonal type accounted for at least 2.5% of the total rates of a sample, and the frequency of the most abundant clonal type is at least 5-fold of the frequency of the fourth most abundant clonal type as a surrogate of the polyclonal background. And the 2.5 cutoff was based on the clinically significant clonal cell population of 5%, and the fact that each clone usually harbors 1 or 2 rearrangements. And the results were interpreted as oligoclonal if 3 or more clonal types had a sequencing rates at least 2.5% of total rates, and the polyclonal or the rest of the scenarios not meeting those criteria. So next, I will use several examples of discordant cases to demonstrate the possible explanations for the discordance and the considerations in the transition from PCR CE-based to NGS-based TRG rearrangement testing. In this first example, CE method detected 1 predominant peak in the V gamma 10 region, while the NGS method detected 2 predominant clonal types. One is consistent with V gamma 10 peak in CE, and the second one corresponds to a rearrangement involving J gamma P over here segment, which was not included in the 2-tube design of the CE assay. And this explains why this clonal type was not detected by the CE test. Therefore, understanding the design of the assay helps to explain the discrepancies seen in the results. In the second example, the CE test detected a distinct peak in the V9 region, while the NGS assay showed a polyclonal pattern. The subject is in remission for the disease of LGL. In a broader sense, on investigation of the reasons underlying the discordant results between CE and NGS assays, we found that 59% of the samples had a clonal CE peak involving V gamma 9 that was not seen as a predominant clonal type by NGS. And 5 of these samples came from subjects with either no history or no ancillary evidence of any hematolymphoid malignancy. Therefore, these findings may be attributable to false positive clonal peaks on CE due to the decreased polyclonal background seen with the low frequency V gamma 9 and V gamma 11 rearrangements in tube B, which has been previously described in literature. Indeed, V gamma 9 and V gamma 11 gene usage, as we saw, was disproportionately low in this cohort. So with this might account for the decrease of polyclonal background and increase the likelihood of seeing spurious clonal peaks on CE. The 4 discordant cases, including this one with the V gamma 9 rearrangements, were further confirmed to be negative by a 1-tube TRG CE method. The NGS assay also effectively addresses this limitation with its 1-tube design, in which spurious rearrangements are less likely to be seen above the more robust polyclonal background, consisting of all possible rearrangements. Okay, so all of the predominant V gamma 9 region peaks seen on CE results were actually found to be less than 1.2% of the total rates of the respective sample detected by NGS. And if we tabulate the NGS rates involving V gamma 9 gene segments only, as shown in the example here, the V gamma 9 clonal peak can be reproduced. The pattern is similar, further supporting false positive clonal peaks on CE due to the decrease of polyclonal background. In the third example, the CE test showed 2 distinct peaks in V gamma 10 and the V9 regions, and 2 clonal types involving the same V gene segments are detected by the NGS assay as 2 dominant clonal types with frequencies over 2.5%. However, the frequencies of the top clonal types did not meet the relatively stringent 5-fold change over background requirement to be called as positive. This is a peripheral blood sample from a patient with a small number of LGL cells. So this is a potentially false negative NGS result, suggesting that further refinement of a clonality interpretation criteria may be needed to improve test sensitivity. In practice, special consideration would be given to these types of cases with interpretations made in the clinical pathologic context. In the last example, the CE assay revealed seemingly 2 distinct peaks within the V128 and the V10 regions, and a weak peak in the V9 region in a peripheral blood sample of a subject with a cytopenia. On the NGS test, However, the predominant peak within the gamma 1 to 8 region was resolved to 3 different clonal types with identical amplicon size. This is an example of a false positive CE result and demonstrated the high resolution advantage of NGS assay to distinguish different clonal types with identical amplicon size. In summary, the NGS-based TRG gene clonality assay has a similar sensitivity and higher specificity compared to a CE-based assay using the established study interpretation criteria. The NGS-based TRG assay provides higher resolution and more objective means of interpretation compared to CE-based TRG assay. The differences in assay design, interpretation criteria, and assay resolution account for the observed discordances between NGS and the CE TRG assay results. The repertoire diversity index could potentially be useful as an ancillary parameter in clarifying ambiguous or difficult to interpret clonality cases. Thank you. This concludes my presentation. I will now transfer the microphone to our second speaker, Dr. Bijal Parikh.
All right. Well, thank you, Melody, for that wonderful talk. What I want to go over, I want to shift my focus to away from the TCR locus and onto the IgG or IG locus. So we've been using NGS clonality methods to determine IG rearrangements for the past 3 years, having retired our capillary-based methods. All right, so here are some of the learning objectives for the talk. But before we discuss our experience, I wanted to go over some of the background of the locus that we're actually interrogating. So as far as the background, immunoglobulins are incredibly diverse, and as a consequence of that diversity, you can imagine with the assay that we're using, if you apply random sampling to those molecules, if you identify a significant number of lymphocytes that share a specific rearrangement, this could either represent a benign reactive process to a foreign antigen, or it could be a more ominous indicator of a potential malignant transformation. So these are things that we need to be aware of. But as far as the IG molecules themselves, the somatic gene rearrangements that occur are sequential, developmentally regulated, and fairly lymphoid specific. The structure of the immunoglobulin molecule is pictured here on the right side, and Melody's already gone over this, so I'm not going to go over this in too much detail, but just to suffice to say that B cells produce membrane-bound molecules, which are B cell receptors, or they can secrete them, which are known as antibody molecules. These are composed of both heavy and light chains, so you could have an IG heavy kappa or an IG heavy lambda here. Where the VJ or the VDJ recombination occurs for the heavy chain, a unique region termed the CDR3 is generated, and this is the most variable of all the CDR1, 2, and 3, also known as the complementarity determining regions. This is what sees the antigen, and what's not shown here in the secondary structure is in the tertiary structure, these CDR regions are really in close proximity to that target antigen. So I mentioned that, if you happen to see a predominant single clone, that could be either benign or a malignant process. So how does a pathologist actually determine whether this is benign or malignant? Well, there are other characteristics that we follow. There are clinical characteristics of the individual that's being tested. There's cytology or histomorphology evidence. There's immunophenotyping by tissue and flow, and there's chromosomal aberrations that are seen in the lymphoid cells in a malignant process. So even after all of that, though, 10 to 15% of these cases of lymphoid proliferative disorders can still remain diagnostically challenging. The other thing I want to point out is that clonality does not equal malignancy. So just because you see a clonal process doesn't mean that you're going down the route of malignancy. And that's important to relay back to anyone who's looking at the interpretation of these results. And finally, even though these are mainly occurring in lymphoid cells, they don't define the lineage. So you could see T-cell rearrangement, TCR rearrangements in B cells, or vice versa. And so you can't use the fact that a rearrangement has occurred to say that this is the lineage of that cell. So those are just some of the caveats or challenges that I want to mention. This is just to show you how complicated the IG locus is, and I'm not going to go over this in too much detail, but suffice to say, not all of the the genes here are functional. For example, for the V genes, there are 38 to 46 functional V genes. These are the open squares in this picture here. There are also D genes and J genes, and all of these combine to give you a final product after somatic rearrangement. What this slide is intended to illustrate is how complex this is, and if you're going to design primers to capture that recombination, you have to be really careful in how you design these primers so that, first of all, it'll capture as much of the diversity as you want it to do, and then that you can multiplex them together so you're not running 15 or 20 different PCR reactions to capture that event. So instead of looking at it this way, here's a more simplified map of the loci. There's the light chains on the top, but I'm going to focus on the heavy chain locus on the bottom. So as Melody mentioned also, this is a sequential process. So for this locus, the D and the J will recombine first, and that'll be followed by the V and the D recombination events. And so when that occurs, you have this final recombined or rearranged receptor, and that's schematized below. And so the various CDRs that I described, the antigenic regions are listed below. So you notice that the V gene contains 2 of these that are encoded in the germline. And then there's a CDR3, which is really unique in that it is it uses the D gene, but also portions of the V and the J. And then there's these nucleotides in between that are encoded that are added by RAG recombinase or TDT. These are sometimes added, or these are typically added in a random fashion. So sometimes the rearrangement is not even productive or functional. What's noted in this picture also is that there's these little arrows underneath these FR regions. These are also called frameworks. So framework 3 is a region where you can design primers. And if you have a short DNA product, that's a really great primer set to use because FFPE tissues tend to have shorter fragments, so you're more likely to be successful. Whereas if you use something like an FR1 or a framework 1 primer, you're going to capture more of that V gene, nearly 70% of it as opposed to 10% with the FR3, and but you need a longer input DNA fragment. When we're looking at somatic hypermutation, we're looking at the actual variants that occur within the V gene. And so that's really difficult to do if you're using an FR3 primer, but much easier to do if you're using FR1, or even if available to you, a leader sequence, which is shown by the indicated by the L here on the picture. So how do we measure clonality? And again, I don't want to belabor this too much because Melody did a really great job of what capillary electrophoresis is. What's important is that these are not quantitative assays, so they have less than ideal sensitivity. So a former mentor of mine referred to this method as akin to reading tea leaves almost. And so this is really unsatisfying for a laboratorian. But what the method did do was introduce the concept of standardization of reagents and reproducibility of results for molecular detection through the biomed group here. So remember, the primers that could be developed needed to be comprehensive and tractable for multiplex PCR. So instead of each lab designing their own set of primers, the Biomed Consortium tested and published a unified set of primers for CE-based clonality testing, along with some interpretation guidelines. And recently, the same group has done, as well as some commercial entities, have developed primers to aid in the standardization of this approach, and this is something we'll discuss a little bit later. As far as the capillary electrophoresis methods, I don't want to go over this in too much detail since we've already gone over for the TCR locus, but what you can see in the bottom are 2 example tracings of a capillary electrophoresis plot. The top is a clonal product, the bottom is a polyclonal product, and you can get products that are in between 2 peaks or multiple peaks. Unfortunately, many of the clinical or the samples that are tested in the clinical lab are not so straightforward, and often NGS testing can be used to help resolve some of these issues. And then to determine somatic hypermutation, really what the lab does is isolate the clonal band from a gel, take that clonal band, sequence it, run it through some blast searches, and then determine how different it is from the germline sequence. This is a really complex, low throughput, error-prone process that NGS can really step in and make a big difference on. So just to restate the obvious advantage of NGS clonality determination, NGS promotes clone identification through sequence rather than size. Although, as Melody showed, size can also be an important supporting piece of evidence as well. So before we jump into the NGS validation that I want to go over, here are a few takeaways as to why we even consider this method. It's a streamlined approach to efficiently detect IGH gene rearrangements. These are highly reproducible and arguably more objective. We can use archival material more easily for these methods because they have shorter fragment sizes. You need very little input DNA. This is something I can speak on a little bit later. And then this is a much easier assessment for somatic hypermutation. And obviously, once you get to that point, you can also look for minimal residual disease testing. So this is an outline of the validation portion of the talk. I'm going to focus mainly on the IGH, so the FR2 and FR3 validation, but the somatic hypermutation with the FR1 underwent a very similar process. So this is not intended to represent the entire validation of the assay, as we need several more hours to go through that process. But in fact, the validation for us took over a year to complete. So in our lab, we have access to the sequencer seen on the right side here. It's an ION S5 sequencer. We have 2 of these, so we validated both of these for the process. Clinical case selection is actually really important. It's a very important aspect of validation. So the question you need to ask yourself is not necessarily limited to what types of malignancies or conditions could I use to test the efficacy of NGS clonality testing, but it should also include what types of specimens am I likely to receive for testing. So in our validation, we chose to directly compare our NGS results to our already established capillary electrophoresis based assay, partly because we had a direct comparator for the assay, and also because we archive all of our DNA samples, so it made it much easier to perform a validation. You see the sample types listed below for IGH, somatic hypermutation. These are typically bone marrow, peripheral blood, but for the standard IGH, not the somatic hypermutation analysis, we had a varied distribution, and we actually have quite a few vitreous samples that we look at, so we wanted to include that in our validation. So it's important to include in your validation the types of samples you will see, in addition to the samples that will, a priori will work well. So here's just a brief overview of the workflow in the lab, and really the most important part here. I've already gone over the specimen types that we accept, and the DNA isolation is pretty straightforward, but the sample prep is really where a lot of optimization needs to occur. Once you get beyond sample prep, sequencing and data analysis are fairly straightforward. So we talked a little bit about sample prep here, which includes a library prep stage first. Certain amount of DNA is added, PCR is performed to add adapters, and then these are removed, assessed on a bioanalyzer to quantify and confirm the size. And then it goes to this middle process here where we prep the template and the chip for loading onto the sequencer. So you have to make these ion sphere particles, enrich the particles, and then load the chip. And this was originally validated on the OneTouch2 instrument. Again, a very labor-intensive process, which resulted in low read counts and high variability. Fortunately, after validation, we were able to acquire an instrument called the Ion Chef, and that allowed us to really increase our read numbers, decrease variability, and the results were that much better. So we looked at other aspects of optimizing our PCR. We wanted to achieve at least 50 nanograms of high quality DNA for this testing. So anything that was greater than 10 nanograms per microliter, we diluted, and anything that was less than we just used straight, and we used 5 microliters of that. But we didn't set an absolute minimum in terms of acceptable DNA, and then there's more on that to follow. We used AmpliTac Gold as a polymerase, and then for every row we wanted to have control. So we have a positive control that we know is clonal, a negative control that we know is polyclonal, and then an NTC or non-template, negative template control. This was really important because we wanted to detect contamination if it were to occur, or if there's barcode hopping, which can happen if there's a really high positive clonal sample on the same chip. We also rotate indexes amongst the controls during and following the validation to examine barcode-specific effects that way as well. So just a brief word on library quantification. We used the bioanalyzer 2100 to ensure equimolar loading of PCR products, so the majority of reads did not come from a single sample, and this is an example of what our tracings look like. The other thing is we used 100 picomolar concentration of our library. How do we choose 100? Well, we actually tried a few different amounts. We saw from 100 to 400, we got very similar results. And then we wanted to understand, could we do this on a 520 chip or a 530 chip? And there are cost savings if we can do this on a smaller chip if we don't have a lot of samples to run. So we wanted to include that in our validation. So here's an example of what we did during our optimization phase to show that both chips functioned well. we took a positive control, ran it 25 times on 2 different instruments, and showed that on average, the percent of our highest rearrangement was very similar, whether we used the 520 chip or the 530 chip. We also did a validation of the different library amounts and didn't see any differences. These were concordant completely for the positive and negative controls for these assays. And so this allowed us to confirm some of the criteria that we initially used to determine what would be a passing QC on these runs. And so as long as their positive control had a read that was over 2.5%, or negative control was under 1%, and those reads were over 20,000 reads, and the negative control had less than 20,000 reads, we consider that a valid run. So we next performed sensitivity determination, and here we obtained clonal control DNA sensitivity panel, a panel from a manufacturer, and tested the different mixtures. And we saw here that while there was some bias, we couldn't get the exact percentage that we expected. In a clinical situation, that exact percentage doesn't matter, and it could be related to some of the limitations of the assay chemistry or the bioinformatics pipeline. But we were able to show overall that a clonal rearrangement of 2.5% total reads with a total read count of 20,000 was consistently detected with all of our dilutions that were at or greater than 2%. And so we use 2.5% as our cutoff for what would be considered clonal versus not clonal. We then took this and did our accuracy study. We took our CE capillary electrophoresis samples, and we determined that we had a very high concordance between capillary electrophoresis and NGS. There were a few that were not positive by FR2, but happened to be positive by FR3. And then there was one sample that just would not show us a clonal result on FR2 or FR3. We did just do some more discrepancy analysis on this specimen. We saw that FR1 gave us a clonal result. So this could indicate that maybe the primer binding sites for FR2 and FR3 were somehow modified, and so we couldn't see them. And so this is another example of why redundancy is also important when you're looking at this. Do you have a backup primer set? And that's why we do FR2 and FR3 together. When we looked at the actual concordance, we saw that the clonal samples agreed almost 96%, but for a polyclonal agreement, we didn't get such a high agreement. So we saw a lot more polyclonal by capillary electrophoresis than we did by NGS. And so this really could represent one of two possibilities. This could be an early clonal process that shows that NGS is more sensitive, or these could be false positives, and NGS, or at least our established interpretation criteria, lack sufficient specificity to discriminate these two. So to adjudicate these possibilities, we would need to look further into additional testing at various time points. But since it wasn't part of our validation, it's not clear which bin these specimens would have fallen into. However, recently published reports really do indicate that NGS is more sensitive. And so these may have been early clonal processes. I just want to briefly touch over, actually, I'm going to skip over the intra-run and inter-run variability because it really does mimic exactly what Melody had shown. But what I do want to go into, and then just come back to our workflow with the data analysis, that's the final step of our workflow, which includes the following steps: demultiplexing, aligning, identifying the genes, merging reads that only differ by 2 or less, calculating the somatic hypermutation rate, and then ranking these. So this can be done either by bioinformatic packages that you design or a vendor supplied software package. Additionally, validation is an ongoing process. It never stops. So when we switched from the one-touch to the chef, we had to do another validation. As we switch hardware and software, validation is required. If there's a new extraction method we're trying out, we need to revalidate. What if we wanted to add FR1 to our traditional FR2, FR3 assessment? How do we probably need to validate that as well. Stereotype determination is important for CLL and for somatic hypermutation. And if you're going to report that, you're going to want to validate that, as well as determining and reporting productive versus non-productive clones. And then if your lab or another lab comes up with interpretation guidelines, it's really important to validate in your sample set, either retrospectively or prospectively, whether they're going to be valid for your situation. I want to briefly go over a case study in which an individual was diagnosed with SLL first, about 4 years earlier. And then he presented with an increasing sense of fatigue, some adenopathy, and lymphadenopathy and back pain. And then he had a inguinal lymph node biopsy that showed a histopathology that was consistent with SLL, CLL. And so clonality, and the important part here is that serum protein electrophoresis was also done. And here there was an M spike shown, and there's an Ig kappa that was demonstrated by immunofixation, whereas the SLL cells were previously shown to be lambda restricted. So the question was, are these two separate processes occurring in the same individual, or are these related? Well, we can use clonality testing to really get at that question. And so when we did clonality assessment from the bone marrow, we saw that the V169 J6 was seen at a 65% level in this individual, but there was also a V18 J4 seen at 4.8%. So mutation rates are very different. Are these really the same process or not? So there could be two processes going on in the bone marrow. Let's go back to the lymph node where the diagnosis occurred. And so, or where the initial diagnosis of SLL was seen. And when we went back to that retroperitoneal lymph node, we only really saw the V169 J6 present at really the same percentage and the same mutation rate. And we didn't see the V18 J4. I'm showing it here, but it falls under our level of what we would call out in a report. So really the process that was occurring in the lymph node was also occurring in the bone marrow, but there was another process occurring in the bone marrow. And so this case shows that you could have coincident SLL and multiple myeloma or plasma cell dyscrasia. Both of these could be arising from the same progenitor cell, or they could represent independent B-cell clones. It's not clear, but we also know, and if you're savvy enough in the field, you know that the V169 J6 has been seen in unmutated CLL, and so it should tip you off that there are just by the nature of what the identity of that V gene was, you could have predicted that this was going to be the outcome. I want to switch lastly for the last few minutes on challenges and opportunities back in this area. So there really are, before we get to challenges, there are two main questions that we need to consider when we're thinking about implementation. Are the advantages of NGS clonality-based testing convincing enough for molecular labs to adopt this technique over capillary electrophoresis-based methods? And secondly, are NGS-based clonality assays standardized enough for widespread adoption? And so to help answer or go through some of those challenges, we have these other issues that we need to work through. So what are the QC metrics that we should be following? What is the gold standard that we should be using? Is it clinical? Is it histopathologic? Is it capillary electrophoresis? Without defining the gold standard, it's really difficult to define the accuracy. How much input DNA should we be using? Does it matter if it's lymphoid rich or not? What about the method of FFPE extraction? And are there certain criteria we should establish to reject samples because they don't meet a specific, size measure? What is the role of repeat testing here? When you repeat something that you're not sure of and the clones change, then maybe that gives you more confidence to say that those are not really clonal in the process of a malignant process, but maybe more of a reactive process. And then how do we decrease turnaround time? Capillary electrophoresis can take 1 to 2 days, but the NGS methodology, at least in our lab, can take 3 to 4 days. So can we stagger this? Do we have enough samples to run this every day or every couple of days? And that's going to be a major challenge. There are other challenges too that I didn't talk about: cost, bioinformatics, and reporting guidelines that are not standardized. But there are opportunities here. So we can use sequence identification, and we can use that to allow for risk stratification. You can use this for MRD monitoring. You can use paired analysis to minimize your indeterminate results. And these guidelines actually are developing from AMP and from other working groups. And here's just a clip from a recent paper in the Journal of Molecular Diagnostics going over how we can start to standardize this approach. So that concludes my presentation. I want to transfer the microphone over to our host at InVivoScribe, Paul McMullen. Thank you for your attention.
We want to thank both Dr. Zhang and Dr. Parikh for their preparation and excellent presentations today. We have now reached our closing time. Please note that we will do our best to take the questions from the audience and provide answers via email. On behalf of InVivoScribe and AMP, we want to thank everyone for your attendance today at this webinar. The session has been recorded and the presentation will be made available via the AMP education website. Thank you. This session is closed.